Thank you for joining the International SeaKeepers Society and our citizen science initiative, the Neuston Net Research Collective. For Dr. Mike Parsons at Florida Gulf Coast University, you'll be collecting sargassum samples from the greater Caribbean area to study its potential to carry toxic single-celled algae, gambardiscus, to coastal areas and affect human health. You'll receive from SeaKeepers and Dr. Parsons the following materials: a styrofoam cooler with ice packs, a 60 milliliter syringe, 25 millimeter filter holder and a petri dish containing 10 filters, a 50 milliliter tube, 5 pre-labeled microcentrifuge tubes, a Ziploc container, tweezers, a 15 milliliter tube with DNA shield and 5 pairs of gloves, pipettes, a waste bag, and a data sheet. To conduct a tow through a sargassum patch, place the plankton net over the side of the boat and use a boom or an arm to hold the net to the side. Collect about a cubic foot of sargassum and record the time and coordinates, latitude and longitude, of the tow. Transfer a portion of the sargassum into the Ziploc container until it is about half full. Add just enough seawater to the container to cover the sargassum and shake vigorously for about a minute. Draw 50 milliliters of water from the container into the syringe. Place the filter in the filter holder, attach the filter holder to the syringe, and push down the plunger to filter the water into a waste bucket or over the side of the boat. There's no need to keep this water. Remove the filter holder and repeat drawing in and filtering water until resistance prevents you from pushing water through the filter. Record the total volume of water filtered. You may filter up to 150 milliliters or more per sample. After filtering, disconnect the syringe from the filter holder, unscrew the filter holder, and carefully fold the filter into quarters with tweezers. Place this filter into the bottom of a microcentrifuge tube. While wearing gloves, pipette DNA shield into the microcentrifuge tube so that the filter is entirely submerged. Place the microcentrifuge tube into the 50 milliliter tube for storage and record the date, time, and sampling location coordinates on the data sheet. At the next sampling site, thoroughly rinse the syringe, Ziploc container, tweezers, and filter holder with seawater before using them again. Try to keep the samples refrigerated or on ice to help with preservation until you are ready to send the samples to Dr. Parsons. For this, you should freeze your ice packs in advance and place them in the box to send. Your work will help the team at Florida Gulf Coast University better understand the contributions of sargassum and gambardiscus to harmful algal blooms and their effects on coastal human and environmental health.